Yes. Yolanda, thank you very much for this kind introduction. Thanks also to Norbert and Gemma for distributing this review, which Katja already introduced. So the ESC was so kind and print a few of them. Unfortunately, they didn't want to, because of environmental reason, didn't want to print for all of you. But I asked them, if you want to have more, just go to the front desk and ask them. They will print more of them. So. Yeah, I would at this point I would also like to thank the organizers for inviting me to give this presentation about the role of calcium signaling in rhythm disorders. During the preparation, I was trying to summarize which are the most important calcium-mediated mechanisms, and I made a list, and I found it's quite a lot. And you heard already about some of them from Katja. You heard about DADs, you heard about EADs, you heard already yesterday about mitochondria and calcium. In addition, calcium can influence the gene transcription and protein expression. It can do this in cardiomyocytes and fibroblasts. And in addition, you have also the L-type calcium and other calcium channels in the myocytes, which can also induce arrhythmias. So I found it quite a challenging task, especially since I was asked to keep it simple and to maybe also be a bit repetitive. So what I did, I summarized all of these mechanisms in three major concepts, which I think play a role more or less in all of these mechanisms. So one is the depolarizing effect of sarcolemmal calcium influx. So this is, for example, mediated via the L-type calcium channel. So calcium influx in the myocytes leads to a depolarization. On the other hand, you have also calcium can directly affect the activity of ion channels or of transporters, which has also already been introduced by Katja. I will give you more detailed insights into that. And also, calcium is a key regulator of protein expression and thereby can also contribute to arrhythmias. I will introduce and guide you through all these three concepts and give you not a complete overview of all the diseases which where this can have an effect, but I would like to select a few examples. And since I'm working on atrial rhythm disorders, mainly on atrial fibrillation since many years, I would like to give you examples from from the atrium, how these mechanisms can contribute to atrial arrhythmias. But the general concepts, which I will introduce, will hold true also for ventricular arrhythmias. So I would like to give you an overview of common principles, how calcium can contribute to arrhythmias. So first of all, the depolarizing effect of sarcolemmal calcium influx. So Katja already nicely introduced the cardiac action potential. I also have a slide for that because I'm a cellular electrophysiologist. You can see here a typical, in this case, an atrial action potential. And this is measured, for example, you have shown here 3 cells. And you have two resting cells and one cell which is excited. So you have like a screenshot of an excitation which propagates through these cells. And in this cell is still resting, and you have an intracellular electrode and an electrode outside, and you can measure in the resting conditions a negative membrane potential. That means the cell internally is negative, and outside you have a positive potential. So, and this is so-called resting membrane potential, which is mainly mediated via potassium channels during the resting phase. And if the excitation now propagates through the tissue, this cell is excited due to the activation of depolarizing inward currents, and the major mediators of this are very fast sodium currents, which are responsible for the upstroke, and of course calcium influx into the cell via L-type calcium channels, which is responsible for the plateau. And during at the end, the membrane potential repolarizes again due to potassium channels. To show you this a bit more direct and to encourage you to play with cellular electrophysiology, I would like to show you and introduce you to a small program where you can do your own cellular experiments let's say in silico. So this is a program which was developed by the Don Burr's lab many years ago, and you can download it here from labheart.org. And I would like to show you how you can do a very simple experiment and how the L-type calcium current contributes to the action potential. So I start that. So you go to start, and then you go to drugs. So you can have a typical cardiac action potential here. You run it, and you also see the L-type calcium current. So you can see that this contributes during the plateau phase. And now we can apply drugs. In this case, nifedipine, a selective L-type calcium channel blocker. So we give it a full dose, apply that, and you can immediately see that the action potential is much, much shorter, and the L-type calcium current is almost done. This is, in this program, it's completely reversible, and you can see again the action potential duration is prolonged. You can also, in this system, you can very easily modulate sodium currents, calcium buffering, potassium currents. You can apply drugs, and you can have you can change the stimulation frequency, whatever you want. I think this is a very nice tool to understand the impact of cellular electrophysiology on the action potential and on the cardiac myocyte. Okay, so I've shown you that L-type calcium currents are important for the prolongation of the action potential. But why is this necessary? This is a very unique thing for cardiac myocytes. And this is necessary in order to prevent a very important arrhythmogenic mechanism, and this is re-entry. I would like to show this here in this little slide and demonstration. So a long action potential prevents the induction of re-entry. Re-entry describes a continuous impulse propagation around the anatomically or functional unexcitable core. And you can see here you have an excited area, and as the excitation travels along this re-entry path, at the end it will hit the initial part, which is still in a refractory phase due to the long refractory period of the action potential. What happens with shorter action potentials? So here you can see that the excitation travels around this circle, but you can see here that the initial part regains the activatability, so it can be reactivated, and so the activation can re-enter this circle, and you can get a circulating excitation, so a re-entry circuit. So in addition to the shortened action potential, slower excitation propagation, a slower conduction velocity can also promote re-entry. So you have this, for example, due to fibrosis. So, and as already introduced by Katja, re-entry is the most important mechanism in atrial fibrillation. So, atrial fibrillation. What? That's true. To sustained atrial fibrillation, because if we take atrial myocytes from patients with chronic, long-term, persistent atrial fibrillation, measure the action potential, we can see that in these myocytes, the action potential is shorter compared to normal sinus rhythm patients. And this assumed that this action potential shortening is a major contributor to the maintenance of the arrhythmia. And it is assumed that the reduction of the L-type calcium current and the increase of the potassium current, IK1, are the major contributors to this shortening, and these are the most accepted hallmarks of atrial remodeling in patients with chronic atrial fibrillation. So I hope I could convince you that direct alterations of the L-type calcium channel or of the calcium influx into the cell can contribute to arrhythmias. So what about the direct effects of calcium on sarcolemmal ion channels and transporters? Before I go into that, I would like to recapitulate the cytosolic calcium handling, which you have already heard a few times, but I was asked to repeat it. And as you all know, the organized contraction of the heart is based on an electrical excitation, which is usually measured in the ECG, and this electrical excitations induces a contraction. So this is scientifically termed excitation-contraction coupling. So what is the major mediator of these excitation-contraction coupling? So we all now know this is, of course, it's calcium. But, of course, this is not known, is known for a long time, but the first person who observed that was actually Sidney Ringer. And I know that you got a few questions yesterday, and Gemma told me that not all of you could answer that, probably also of spelling mistakes. And but I would like to give you the story behind it, which also helps to understand calcium handling a bit. So in this publication from 1882, Sidney Ringer observed that if he explanted a frog heart at room temperature, it still remains contracting even if you wash out the blood and apply a sodium chloride solution. If you apply a sodium chloride solution, he was able to maintain the contraction of a frog heart at room temperature. You will now ask, how is that possible? How is it possible just to have a physiological sodium chloride solution to maintain beating of a heart? If you try that in your lab, it will probably not be possible. And Sidney Ringer experienced the same. He tried after the publication himself, and what he found out, it doesn't work. And so he went back, and the problem is that his technician was doing the experiment, and he found that she did an important difference, what would probably not happen these days anymore in the lab. And so he had to retract this paper with the following comment. I discovered that saline solution, which I had used, had not been prepared with distilled water, but with pipe water. I at once tested the action of saline solution made with distilled water, and I found that I did not get the effects described in the paper referred to. It is obvious, therefore, that the effects I had obtained are due to some of the inorganic constituents of the pipe water. And he figured out, after testing, that calcium is one of the most important contents of pipe water, which contributed to the beating of the heart. And after he added calcium, he could maintain the beating of the frog heart. So this was the first observation that calcium is very important for the contractile function. And at this time I would also like to acknowledge that I have taken all this information from this nice little review from David Eisner. He also presents this on a YouTube video, and I really highly recommend you to read that paper. It gives you a lot of background information, a lot of interesting little things, and you can also recommend you to watch this little YouTube video from David Eisner. So we now know that calcium is the major mediator in the excitation-contraction coupling process. And we can recapitulate this on a single atrial myocyte. So you can see here a myocyte isolated from a patient undergoing open heart surgery for coronary bypass grafting. So we took the samples, isolated atrial myocytes, and in order to stimulate them electrically, we used the patch clamp technique. So you can see here the patch clamp technique, the pipette, and we applied the depolarizing voltage pulse. And in response to this voltage pulse, you can see here the activation of the L-type calcium current. And you can also nicely see that in response to this calcium current, you have a cellular contraction, which we can also quantify here. So calcium is supposed to be the major mediator, and if we look a bit more into the details, we see that the influx through the L-type calcium channel activates a much larger calcium release from the sarcoplasmic reticulum, and the calcium binds to contractile filaments and initiates contraction. During diastole, the calcium is pumped back into the SR via SERCA or sodium-calcium exchanger, and so we get back to normal calcium levels. So in order to measure this or image that experimentally, we loaded the myocyte with a calcium fluorescent dye, fluo3, and you can see that the green lightening up of the cell reflects the systolic calcium transient, and the decay is the calcium removal from the cytosol. So we did these experiments in patients with sinus rhythm and chronic atrial fibrillation. And as I mentioned before, we found, as many people before us, that we have a reduced L-type calcium current amplitude, which contributes to the maintenance of AF. And this smaller L-type calcium amplitude triggered a much smaller calcium release from the sarcoplasmic reticulum, as you can see here. So this was, to be honest, not that surprising. But what we found more interesting was that the diastolic calcium levels were higher in chronic atrial fibrillation. So we thought, like, let's have a closer look into the diastolic calcium level. Here you can see, and this is to illustrate these measurements. This is actually a ventricular myocyte loaded with flow 3, a calcium indicator, but this is not stimulated. It's a resting cardiomyocyte. And you can see many of these little flashing dots here. And these are supposed to represent spontaneous ryanodine receptor openings during diastole, causing small spontaneous calcium releases, so-called calcium sparks. And if these sparks accumulate locally, they can initiate so-called calcium waves, which travel through the whole myocyte. And these calcium waves can initiate arrhythmias. So Katja already introduced it very nicely. The calcium released through the diastole is removed from the cytosol via the sodium-calcium exchanger, which brings 3 sodium ions into the cell per calcium ion extruded. So this, as Katja already introduced, is an electrogenic process which can depolarize the membrane potential during diastole and called so-called delayed after depolarizations. So, and if these delayed after depolarizations are large enough, they can trigger a new action potential and initiate arrhythmias. So in this study, we supposed that, or we hypothesized that DADs can contribute to arrhythmias. So I would like to come back to the figure we had before. So electrical excitation does not only induce contraction mediated via calcium, but as I've just shown you in the figure before, calcium can also induce electrical excitation itself. And this is called reverse excitation-contraction coupling. And now here's the example I was talking about. We were interested in whether spontaneous calcium releases and DADs occur more frequently in patients with chronic atrial fibrillation. And in order to investigate that on a cellular level, we measured simultaneously calcium concentration and membrane potential with the patch clamp technique. And you can see here, after steady state stimulation, we stopped the stimulation, and already in sinus rhythm you can see here very little small calcium releases together with associated very small membrane depolarizations, which are called DADs. In atrial fibrillation, these spontaneous calcium releases occurred much more myocytes, and they occurred with a much higher frequency. And we quantified that and found that the calcium spontaneous calcium release amplitude was not so much higher, but the membrane potential change was significantly higher. So in other words, the same calcium release created a much higher membrane depolarization in atrial fibrillation. And in addition, these spontaneous calcium releases occurred much more frequently, so we assume that these could contribute to the maintenance of atrial fibrillation. But as I've told you, this mechanism of spontaneous calcium release and delayed after depolarization does not only contribute to atrial arrhythmias, but also to, as already introduced by Katja, catecholaminergic polymorphic ventricular tachycardia, or CPVT, and this is a common concept which holds true in several arrhythmia mechanisms. I would like to introduce to you a second mechanism which I was working on during the last months and almost years already, and that's what is related to a question which you also got yesterday. So, but first of all, some background information. I introduced to you that into chronic atrial fibrillation is based that somehow the shortening of the action potential plays a role, also that delayed after depolarizations may play a role in the maintenance of atrial fibrillation. But we were wondering, a major risk factor of atrial fibrillation is heart failure. And we were wondering, does heart failure in the atrium induce alterations in the atrium which may predispose atrial myocytes to develop these arrhythmias? So, and as I introduced to you, calcium handling is important for arrhythmias. We looked into calcium handling in these myocytes. So these is a study in atrial myocytes from patients with and without heart failure, but without atrial fibrillation. So they were all in sinus rhythm during the study. So the first thing we found is that the L-type calcium current was not that much different in heart failure and non-failing hearts. But when we looked on the calcium transient amplitude, we were surprised because the amplitude was largely reduced in atrial myocytes from patients with heart failure. And we were puzzled about this because the triggered calcium was unchanged, but resulted in a reduced systolic calcium transient amplitude. In order to understand this, we wanted to investigate the SR calcium load, so the calcium content of the sarcoplasmic reticulum, because it is known that a reduction of the SR load can contribute to the reduced calcium amplitude, even if L-type calcium current is unchanged. So we studied the calcium load by a very standard protocol, which is very well established. So we applied caffeine. Caffeine, caffeine is from coffee, and what it does, it is if you apply very high concentration, 10 millimolars, it leads to an almost complete depletion of the sarcoplasmic reticulum from calcium. And this is reflected by the caffeine-induced calcium transient. So keep in mind that I showed you the systolic transients, which had an amplitude of around 200-300 nanomolar, and the caffeine transient is much higher, and it is an accepted index for the SR calcium load. So at the same time, when we apply caffeine, the removed calcium, released calcium is removed from the cytosol via sodium-calcium exchanger, which is, as mentioned already several times, is electrogenic. And in response to this caffeine-induced calcium transient, we could measure a depolarizing inward current in response to the caffeine-induced calcium transient. So, and the area under this curve is an accepted index of the SR calcium load in these myocytes. So we investigated both indices in heart failure and non-failing atrial myocytes, and what we found was a discrepancy. So the amplitude of the caffeine transient was reduced, and the total calcium was unchanged. So it took me quite a while to understand this, and I had to go back into a famous book from Donald Burrs about excitation-contraction coupling. And I found what I was not really, I was aware of it, but I didn't take it into consideration, that most of the calcium released during the systole is bound to calcium buffers. So at least it is I made this in proportion. 99% of the systolic calcium released from the sarcoplasmic reticulum is bound to calcium buffers, and only 1%, you see this little ball here, only 1% is free and can be measured via the calcium indicators. So in other words, I showed you that around 100 nanomolar is what we measure, but in fact around 100 micromolars is released during the systole. So, and this explains why, it could explain this difference, because this measures actually the free calcium, and the integrated NCX measures the total calcium release during the caffeine puff. So, and we used, to quantify this, called buffering, we used the protocol which was established by Andy Trafford and David Eisner. So we plotted this integral, this area, during the decay of the calcium transient versus time. So we have a measure of the total calcium during the time, and then we plotted the total calcium versus the free calcium. So, and if you do that, we can fit a Michaelis-Menten curve, which is a typical buffer curve with the B max as a measure for the total buffer volume, and the KD value is a measure for the calcium affinity of the calcium buffers. And the reduction in this key value indicates a higher affinity of the calcium buffers in these myocytes. So in the next step, so these are preliminary data, but in the next step we were wondering, okay, what are these buffers? What could contribute to the increased buffering in myocytes from patients with heart failure? And I got help from a recent publication from Gottfried Smith and David Eisner, also I can really highly recommend to read that. And I found they describe that major buffers in the cell are SERCA and troponin C. Again, the free calcium is only a very small contribution. So we measured the SERCA, or we estimated SERCA activity by analyzing the decay of the systolic calcium transient. And because the systolic calcium transient is about 60 to 70% due to calcium removal from the sarcoplasmic reticulum, from the cytosol. So we found that this is even slower compared to non-failing hearts. And together with the unchanged total SR load, we excluded that an increased SERCA activity contributes to the increased buffering. But what is with the with the contractile filaments? So here I got help from the lab of Christoph Mark and his colleagues in the cardiac surgery in Würzburg, because they do regularly skinned fiber experiments in atrial trabecula from patients undergoing open heart surgery. And if you and so we substratified their experiments, and you can see leftward shift of the calcium curve, indicating again a higher calcium affinity of these filaments. By the way, this is a similar technique which Jolanda van der Velde is an expert in, and she introduced that already yesterday. So this points suggest that a higher affinity of the contractile proteins may contribute to the buffering in the atrial myocytes. So can this contribute? Can a high buffering contribute to arrhythmias? So this question was answered, or was tried to answer by the group of Björn Nollmann a few years ago, where they investigated mouse hearts from transgenic mice with different affinity of the calcium proteins. And you can see a leftward shift of the curve, and they showed that mice with a higher calcium affinity of contractile filaments have a higher susceptibility to pacing-induced ventricular arrhythmias. So, and the mechanism they proposed, which is also proposed from other groups, is that because of the higher buffering, SERCA cannot pump back the calcium from the cytosol into the SR, so it cannot completely refill the SR, so that the next calcium transient is smaller. So you get a so-called calcium amplitude alteration, or calcium alternance, and now mediated via calcium-sensitive ion channels or transporters, you can get a so-called action potential alternance, which with long and short action potentials, and this contributes to a kind of tissue heterogeneity and contributes to the maintenance of arrhythmias and contributes to the occurrence of reentry. So we were wondering whether we can induce this in single cells, and in Göttingen, these are again preliminary experiments, but we obtained iPS cardiomyocytes from my colleague Antje Ebert, and she CRISPRed a mutation in these cells to induce a higher calcium affinity. So this mutation is known to be associated with dilative cardiomyopathy, and when we investigated these cells, we found a more or less similar phenotype as we found in the heart failure patients. We found even an increased L-type calcium current, but a reduced calcium transient amplitude and a slower tau value. And if we analyzed buffering again, we found the discrepancy between reduced caffeine-induced calcium transient and higher integrated NCX current. And again, quantifying this buffering, we found a higher affinity of the calcium buffers in these cells. So can we now induce alternance in these cells? And this is an experiment which we did actually last week. It was done by a MD student, Philip Jung. And what he did, he paced these cells at different frequencies from 0.5 to 5 hertz. And you can see this even in control cells. You can see that they follow very nicely at lower frequencies, but at 5 hertz you can see that they do not capture every stimulation. And you can see that the second stimulation results in a much smaller calcium transient. And what happens now in the mutated cell? So more or less all of these cells at a certain frequency develop these alternants. There was only one cell which did not develop alternants in the control group. But in the mutated cells with the higher calcium affinity, we could show that there was a much lower frequency necessary to induce alternance in these cells. So these had a much higher susceptibility to calcium alternance, and we think this may be a mechanism which also can contribute to atrial arrhythmias in heart failure patients, and we will study this in the next years. So briefly, I would like to come to the last point because I didn't want to take this out. Calcium is a key regulator of protein expression. And exemplarily, I would like to introduce to you two excellent studies from the group of Stan Natel, who showed that calcium itself contributes to the downregulation of the L-type calcium current and the upregulation of IK1 current, which I told you are hallmark of atrial remodeling in patients with chronic atrial fibrillation. So what he showed is that I will just show you a schematic of this principle, that because of the high frequency in atrial fibrillation, the tachycardia, you have a much higher calcium influx. This leads to a higher calcium concentration in the cell and activates the calcium-dependent phosphatase calcineurin, and this activates the NFAT. NFAT, or dephosphorylates NFAT. NFAT is well known in immunology, because it's the nuclear factor of activated T lymphocytes, but it's obviously a common principle. And the dephosphorylation leads to a translocation of NFAT into the nucleus, where it regulates gene transcription and basically leads to a downregulation of the mRNA of the L-type calcium channel. But at the same time, it leads to a downregulation of inhibitory microRNAs and thereby to an increase of the expression of IK1 channels. So he concluded that the same system that controls L-type calcium downregulation in AF also controls the upregulation of IK1. And so maybe calcineurin and NFAT and maybe this microRNA could be interesting targets in AF. So in order to summarize, I have hopefully convinced you that calcium handling can contribute to arrhythmias via its depolarizing effect of calcium influx into the myocyte. Calcium can also modulate ion channels or transporters and thereby contribute to alternance or to DADs. And also calcium is a key regulator of protein expression, and this is termed excitation-transcription coupling. And keep in mind that this not only happens in atrial myocytes or ventricular myocytes, but could also contribute via modulation of cardiac fibroblasts. At the end of this presentation, I would like to thank my group. I'm in Goettingen since 2.5 years, and these people here, PhDs and MD students, and also Fleur Mason as a postdoc, spent a lot of efforts to establish the technique. Some of them I showed you in the new group, and they did really hard work to establish the group and to start with running the lab. I would like to thank our collaborators, and I would like to thank you for your attention. Thank you very much.
Thank you, Niels. I like it that you link the electrical signal to the myofilament. So that's questions. I have a question. I was wondering, you put the heart failure groups into high and low ejection fraction. What's the non-failing? Is that diastole? Is that HFpEF then?
No, this
Or
is definitely. So we looked for the heart failure. We wanted to make sure these are really non-failing heart with normal ejection fraction, and they have also no diastolic dysfunction,
okay,
and the other one have a very low ejection fraction. So we also wanted to make sure that we really see a phenotype. So that's why we selected patients with an EF lower than 45.
How do you get, during what surgery do you get the samples then?
So these are coronary artery disease patients usually. Which either have heart failure because they had probably an infarction before, or because of other reasons. But these are basic. Not all of the operated patients have heart failure.
No, okay, yeah. I was just wondering. And what's
My name is Alina from Ukraine. I want to ask: have you studied mitochondria, the role of mitochondria in this calcium handling in cardiomyocytes, and also what is the concentration of calcium accumulate in these organelles?
So this is an excellent question. We are actually working on this. So we have a project where a PhD student together with Fleur Mason and Julius Pronto, they are extensively working on that, and we're collaborating with Christoph Mark on this to establish techniques where we can simultaneously measure calcium and in the mitochondria and in the cytosol. It's very, very difficult to quantify it in the mitochondria. So based on recent evidence also from John Lederer, I don't think that there's a really strong increase in the calcium or accumulation in the calcium in the mitochondria. I don't think mitochondria contribute very much to calcium buffering. But I think that the calcium contributes to regulation of mitochondrial activity, and we think that the interaction between SR and mitochondria may be disturbed in atrial fibrillation. But if you look in PubMed and atrial fibrillation and on what is known about that, you get around 20, 30 papers during at all. So there's very little known about mitochondria and atrial fibrillation, and we really much look into that and try to figure out what's going on there.
Thanks.
So what's the cause for the high calcium sensitivity? What's the which change that the myofilaments is introduced to?
So we could basically only speculate. It could be that there are neurohumoral factors which are also are used to adapt the ventricle to the heart failure, also affect the atrium. It could be stretch. So we currently investigate the effect of stretch on the atrium by using, together with Wolfram Zimmermann, to generate engineered heart muscle and stretch those chronically and see whether they develop similar phenotype or higher calcium affinity. But we have actually, it was just an observation we had, and we're just going on, and I wanted to share this data with you because I found the buffering very important.
It is, yes. Okay, so if there are no more questions, thank you very much for a beautiful session.